Hello students, welcome to another lecture on Understanding Biology chapter 31, the living plant. The student learning outcomes for this lecture is to being able to describe the conditions that produce root pressure, explain how guard cells regulate the opening of stomata, describe how plants are adapted to reduce water loss, describe how plants are adapted to high salt concentration. In this chapter, we are going to learn about how water is transported through xylem from root to shoot. Water and dissolved solutes move from root cells through the endodermis to the tracheids and vascular members of the xylem. We did learn about this in our previous lecture. So roots actually continuously absorb water and ions from soil, which change the tonicity of the root cells. It actually increases. As you remember, tonicity is the capability of a solution to modify the volume of cells by altering their water content. This increased tonicity movement of water actually create movement of water into the root cells, and water potential and turgor pressure actually increase in these root cells. So now you kind of get the idea how roots actually able to keep absorbing water because of the tonicity and the water potential. We did learn about water potential in our previous lecture, so if you don't remember, please go back to that lecture and refresh your memories about water potential. So now the idea is to look at how water can move into the xylem and from xylem up in the plant. So first we need to understand that we need to understand root pressure. So root pressure is caused by the accumulation of ions in the roots. At times when transpiration from the leaves is very low or absent, this root pressure itself is enough to transport water and mineral into not that much heights but certain heights. So at night especially, root pressure can increase. This accumulation is result in increase of iron concentration within these root cells. This actually, when the iron concentration is increasing, this draws more water into the roots by osmosis. As you remember, water actually moves from a high water concentration to a low water concentration. So when the iron concentration is increasing in those root cells, they have low water concentration. So water will move from soil into the roots because of the water potential. This results more water in the roots, and actually it will create a gradient that will result in movement of water into the plant and up to the xylem. Even in the absence of transpiration from stomata through the leaves, root pressure is, when the root pressure is very high, it can force water up to the leaves. This water can get lost in the process of guttation, but guttation cannot move water up long distance. This process is called guttation actually. So root pressure alone, it's actually not enough to explain the xylem transport. So now we are going to look at actually what is driving the xylem transportation. So transpiration actually provides the main force for moving water and dissolved solutes from roots to leaves. Water moves from soil into the plant only if water potential of the soil is greater than that of the root. Too much fertilizer or drought conditions can lower the water potential in soil and limit the water uptake. Water in a plant move along a water potential gradient from the soil where water potential is 0 to more negative water potential in roots. Stems, leaves and atmosphere also have negative water potential. So evaporation of water in a leaf creates a negative pressure or a tension in the xylem. This actually pulls water up to the stem from roots. The strong pressure gradient between leaves and the atmosphere cannot be explained by evaporation only. It actually depends on, as water diffusing from the xylem of the leaf, it forms a thin film of water along in the mesophyll cell walls. If the surface of this air-water interface is smooth, the water potential is higher than if it is rippled. The driving force of transpiration is the humidity gradient inside the leaf, because inside the leaf has more humidity than the humidity outside of stomata. As the rate of evaporation increases, diffusion is insufficient. So this film of water get rippled and create the pull on the column of water in the xylem. This is what actually draws the transportation in the xylem. So you need to explain, if someone asks how xylem transportation happens, it actually happens because of the evaporation and transpiration, as well as the humidity, variation in humidity inside the leaf as well as outside of the stomata. That thin film of water actually get rippled, and then it drives the pull of column of water from the xylem into the leaves. That actually help the xylem transportation. So let's look into water's inherent tensile strength. Tensile strength of water rises from the cohesion of water molecules and the ability to form hydrogen bonds with one another. Water has so many qualities, so we are going to talk about a couple of them in here. So one is that ability of water molecules to form these hydrogen bonds with one another. This actually help them to attach to each other through cohesion forces. This is the basis of cohesion tension theory of the bulk flow of water in the xylem. The tensile strength of a column of water can vary depending on the diameter of the column. As you know, tracheids and vessels are tiny in diameter, and they have very strong cohesive force against the gravity. Further, we did talk about adhesive forces. The adhesive forces is actually the ability of water molecules to attach to the side of the tracheid or xylem vessels. Actually, this will further stabilize the column of water against the gravity in the xylem. So the tensile strength of a water column can be usually, if it's strong if it is not broken, but air bubbles can make this tensile strength to be broken. Usually air bubbles get introduced into the column when a vessel is broken, or sometimes any other accident can happen where air bubble can introduce into these tubes. This can break the column and continuity of the water column, and cohesion will fail. This can expand and block the whole tracheid in the process called cavitation. Cavitation can stop water transport and lead to dehydration. So there are certain anatomical adaptations to minimize the cavitation. The one is that the presence of alternative pathways that they can create a water column to avoid these air bubbles, and individual tracheids or vessels are connected to each other via pits. These actually can prevent air bubbles getting larger and prevent them passing through these pits. So the damage can be minimized actually. So let's look into bulk flow of water and minerals through xylem. So we know that tracheids and vessels are not only transporting water, they are essential for the bulk transport of minerals. Minerals are actively transported into the roots and move through the xylem to other metabolically active places of the plant. In xylem, you can find phosphorus, potassium, nitrogen, iron abundantly. Calcium is an actually essential nutrient that cannot be transported elsewhere once they have been deposited. So bulk flow actually include not only water but also other minerals. So more than 90% of water taken by the root of a plant is ultimately lost to the atmosphere from the leaves. Photosynthesis actually require a supply of carbon dioxide from atmosphere. In order to reduce the loss of water to atmosphere, plant can actually reduce by by closing stomata. But when stomata are closed, you cannot absorb carbon dioxide. So at half of the stomata has to be kept open to support photosynthesis. What happens when you close stomata? They actually vapor pressure gradient is less, so you would lose less water. But this is only safe for short-term basis because, like I mentioned, you need to have supply of carbon dioxide for photosynthesis. Now let's see how turgor pressure in guard cells can cause stomata to open. So guard cells are the only epidermal cells that contain chloroplast. You know about guard cells from our previous lectures. Guard cells have been shaped like kidney beans, and they contain distinctive wall construction which is thicker on the inside and thinner in elsewhere. When these turgid, they will actually bulge out and bow, causing them to open. So that is very important about guard cells. They can acute uptake of this potassium chloride and malate ions, develop this turgor in the guard cells. This will increase the solute concentration of these guard cells and decrease the water potential. When water enters through osmosis, these guard cells become turgid and then they will open stomata. So you can see that when turgor is introduced, because of the ion concentration, these stomata will open. Now you know that how guard cells involve in opening stomata. In many plant species, guard cells regularly become turgid in the morning when photosynthesis occurs. Actually, they lose their turgor during the evening time due to the sucrose accumulation via photosynthesis. I mentioned earlier that these guard cells are the only epidermal cells that have the green pigment, chlorophyll in them. So they do conduct photosynthesis throughout the day. So when they produce sucrose, the active pumping of sucrose out of the guard cells lead them to lose their turgor and close up the stomata in the evening usually. So environmental factors can affect the rate of transpiration. Transpiration rates increase when the temperature, wind velocity, these kind of environmental factors can affect the transpiration rate due to the rapid rate of water evaporation. There are several pathways to regulate stomatal opening and closing. The abscisic acid is a plant hormone that we will talk in future that plays an important role in allowing potassium ion to pass rapidly out of the guard cells and close these stomata during the drought condition. So abscisic acids actually can bind to receptors in the plasma membrane of guard cells that trigger signaling pathways to open potassium and chlorine and malate ion channels that will reduce the turgor pressure, followed by the water loss and closure of the guard cells. Another pathway to regular stomata is by using carbon dioxide concentration, light and temperature. When carbon dioxide concentration increases, guard cells of many plant species are triggered to decrease the stomatal opening. Also, when light intensity is increasing, the temperature of leaf rises. This will trigger the opening of stomata to cool the surface of the leaf. Stomata may also close when temperature exceeds 30 to 34 Celsius and water stress is high. Stomata will only open at night when the temperature is cooled. Now that we know stomata opening and closing is coordinated with the environment, let's look at how plants are adapted to water stress. You know that plants are anchored to the soil via their roots, and they cannot relocate when water availability or salt concentration changes. Therefore, the plants will adapt to their environment conditions that are critical to their survival, such as salt concentration and drought. There are many mechanisms for controlling the rate of water loss. Immediate response to water loss is to regulate opening and closing stomata, but there are many morphological adaptations that provide long-term solution to drought. Here are some examples. Some plants become dormant during the dry times of the year. They limit transpiration by shedding their leaves, such as deciduous plants. Having thick, hard leaves with few stomata on the lower side is also helpful. Leaves covered with masses of woolly-looking trichomes prevent water loss, and having stomata inside pits actually help to reduce the water loss. And that are some adaptation for drought. But what about flooding? Another environmental condition that plant has to face sometime is flooding. Receiving too much water can deplete availability of oxygen in soil and interferes with the transport of mineral and carbs in roots. Oxygen depletion can lead to decreased cellular respiration. Plants can respond to flooding by forming larger lenticels, which facilitate gas exchange, and they can also form adventitious roots that reach above the flood level for gas exchange. So we can actually, plant also have adapted to life in fresh water from forming parenchyma, which lose parenchymal tissue with large air spaces, and they collect oxygen and transport it to the submerged part of the plant, which are some of the adaptation. Plants such as mangroves actually grows in areas normally flooded with salt water. These plants have parts that are submerged that need oxygen and also need controlling of the salt balance. In order to balance salt in these plants, they actually secrete or dilute salt as it enters to the plant. Black mangroves have long, spongy, air-filled roots that emerge above the mud. These roots are actually called pneumatopores. They have lenticels that allow the air change for the submerged part of the plant, and their succulent leaves can store a lot of water to dilute salt. They also can secrete salt and block salt uptake at root level. So these are some of the adaptations that these mangrove plants have to tolerate salt. So we did talk about how plants are adapted to drought, how plants are adapted to flooding, and how plants are adapted to salt concentration. So in summary, stomatal opening is controlled by guard cells, which change the shape based on the water content. Abscisic acid controls the movement of potassium ions, which affect osmosis. Stomata close when plant is under water stress. Adaptation to drought include dormancy, leaf loss, and leaves that minimize the water loss. When plants are flooding, oxygen deprivation leads to lower cellular respiration. If a plant is exposed to salt environment, it may exclude salt from uptake, secrete it after it has been taken up, or dilute it. I hope you enjoy this lecture. Until we meet from another, have a good day. Thank you.